So in this experiment, we're going to look at the IMViC, and this would be activity 5-4, 520, and 5-9. And so we're going to start out talking about day one and how we would set up this experiment first. So what we would do is in groups of two, so in pairs, what we would have you do is each person would get 3 tubes. They would each get 1 tube with a red cap. That is an MRVP broth. They would get 1 tube with a green cap. That is for the indole test. And they would get 1 citrate slant. And so, for example, let's say that I was assigned to do E. coli. I would inoculate E. coli into all 3 tubes. So I would inoculate it into an MRVP broth, into an indole, and into a citrate. And then my partner would inoculate Enterobacter aerogenes, which we would abbreviate EA, into an MRVP broth, an indole broth, and a citrate slant. And so we would use a loop to inoculate the broths, and we would use a needle to inoculate the citrate slant. Now, normally in the past, when we have inoculated a citrate slant, we would do a zigzag inoculation using a loop. However, in this particular experiment, we need to make sure that we don't transfer too much bacteria. So what we're going to do is we're going to use a needle because, again, we don't want to transfer too much. And you'll see why when we talk about the readout. So you and your partner would make these tubes and get them set up. The citrate tubes are screw top caps, so we have to make sure that the caps are on loose before we put them in the incubator. And so we would set up these 6 tubes, then we would put them in a can, put them in our incubator. We would incubate at 37 degrees Celsius, so human body temperature, for 48 hours. And after 48 hours, then we would do the readout. So now I have a video demonstrating the setup for these tubes. So in this experiment, I'm going to demonstrate how to set up our IMViC. IMViC stands for indole. So we have an indole production test. We have the MR, methyl red. V, Vogue's Proscour, that's in the same broth. This is an MRVP broth. And then the C is citrate. This is a citrate slant. We're looking at citrate utilization. So when we inoculate an IMViC, there are 3 tubes that we need to inoculate. So we have the green one, which is called a tryptophan broth. Again, that's going to be used for the indole test. The MR and VP are both in this MRVP broth. So this is both in one. And then we have our citrate, which is our slant. So when we do this, we would have 3 tubes per organism. So we would have 3 tubes that have E. coli. This is our E. coli that we're using. We would also have 3 tubes that have our Enterobacter aerogenes, which is our other organism that we're testing. Again, for the purpose of this, I'm just going to demonstrate setting up 1 set of tubes, meaning just for 1 organism. But then you have to imagine that there's going to be 2 sets. So you would have in your rack, if you were doing this, there would be 6 tubes total. So when I go to inoculate the 2 broths, the green cap and the red cap, which tool am I going to use to inoculate those? And the answer is we want to use a loop, because when we inoculate a broth, we want to use our loop. For our citrate is on a slant. Normally when we inoculate a slant, we use a loop. However, the citrate test is a little bit different. In the citrate test, we want to only inoculate lightly, because if we have a heavy inoculation, it's going to affect our results. I'll explain why later. So when we inoculate a citrate slant, we are going to do so using a needle. So we're not going to do our zigzag inoculation with a loop. Instead, we're going to do it with a needle so that we don't pick up too much bacteria. So let's start with the broths first. So I'm going to start with my loop, and I'm going to flame sterilize. And I'm going to let it cool. Again, I have my E. coli here. So palm side up, pinky ring finger, flame it, and I'm going to go in, and I'm just going to pick up some of the bacteria. Come out, flame sterilize, set that down for a minute. I'm going to inoculate my indole first. Flame it, just swirl my loop in the broth. Flame it, cap back on. I'm going to move it in my rack so that I know that that's inoculated. I'm going to flame sterilize in between my tubes. So before I go back into my slant, I'm going to flame sterilize and let it cool. So again, pinky ring finger, hold the cap between your hands, your fingers, flame it. Go in, pick up some bacteria, flame it, cap back on. I'm going to inoculate my MRBP broth. So cap comes off, flame it, swirl it in the broth. Come back out, flame, and flame sterilize my loop. And then I can set my loop down. I'm done with my loop. I'm going to inoculate the citrate slant, remember, with a needle. So I pick up my needle, and I'm going to flame my needle, and I'm going to let it cool. Now, once this cools, I'm going to pick up bacteria. So I'm still going to inoculate my citrate slant using a zigzag inoculation. However, when I do that, I'm not going to stab inoculate. So not like in the OF test where it's a stab inoculation. It's still a zigzag inoculation, but it's using a needle. So I'm going to take my tube, flame it, go in and pick up some bacteria, flame it, I'm going to take my citrate slant, I'm going to flame it, and I'm going to do my zigzag inoculation using my needle. So starting at the bottom, zigzag and work my way up, flame, cap, and then I need to flame sterilize my needle before I set it down. Now, notice for my indole and my MRVP, these are loose caps. The citrate, though, this is a screw top cap. So the way that we put these lids on really matters for the screw tops. I can't have it all the way tight. If I do that and air can't get in, then there's no way I'm going to see any growth. That's going to affect my result. So you have to make sure that when you use these screw tops, what I do is I do put the lid on all the way, and then I give it about a half turn loose. You want it to be able to wiggle, but not to fall off. So you want your cap loose. It's not going to fall off, but air can still get in. And so I would do my 3 tubes for my organism, and then I would do the other 3 tubes with my second organism, and then I would take these tubes and I would put them in the incubator, and then I would do my readout 48 hours later. And so this is how I set up my IMViC. So now we're going to move on to day 2, talking about our readout of our IMViC tubes. So after our tubes have been incubated for 48 hours. So for our IMViC, this is the second most important ID procedure after the gram stain, right? And so recall that about 95% of all medically important bacteria are either gram positive or gram negative. And so a quick way to determine something about the bacteria would be to do a gram stain, because again, that would help you decide on an appropriate antibiotic for a patient that has an infection. Now, if we needed to narrow it down further, then we could do an IMViC. And that would help us to narrow down what bacteria we might be looking at. So when we do our IMViC, the I, the first I, stands for indole. This is an indole production test. This is a test looking at to see if indole is produced. The M is for methyl red. So the methyl red is in the MRVP broth. It actually is used for both the M and the V test. The V is for Voges-Proskauer. So these are two tests that are looking at the way that glucose is metabolized by bacteria. The little I is just there to make it easier to say. It doesn't actually stand for anything. And the C stands for citrate, for citrate utilization, to see if citrate is utilized as a food source by the bacteria. And so these are the tests that we have. So for our MRVP, the name of the media is called simply MRVP broth. So when you're filling out your biochemical sheet, the name of the media that you're going to put for your MR test and also for your VP test would be MRVP broth. For the indole test, the name of the medium that you're going to put on your biochemical identification sheet would be a tryptophan broth. These are referred to as a tryptophan broth. And then lastly, for the C, there would be a citrate slant or a Simmons citrate agar slant. Now, when you're filling out your IMViCs, my recommendation would be to make 4 separate biochemical sheets. I would have one that's separate for indole. I would have one for methyl red. I would have one for Voges-Proskauer. And I would have one for citrate. It's going to help you when you study if you put those on 4 separate sheets. If you try and fit it all on one, you're going to get confused. So put it on 4 separate sheets. So remember that when this experiment was set up, there were 2 types of bacteria. We have E. coli. That would be the tube we labeled EC. And so E. coli would be inoculated into an MRVP broth. It would be inoculated into a tryptophan broth. And it would be inoculated into a citrate slant. And then we would have Enterobacter aerogenes. And that would be the tube that's labeled EA. And again, we would have inoculated an MRVP broth. We would have inoculated a tryptophan broth. And we would have inoculated a citrate slant. And so now we need to talk about how do we do the readout to see which of these tests are positive or negative. So for our citrate test, there's nothing we have to add to that tube. All we're going to do on this test is simply observe the tubes for growth and color. So we're going to see, do we see growth on the slant? And then what is the color of the medium? Is it green or is it blue? To the indole test, so to our tryptophan broths, we would need to add 7 drops of our Kovacs reagent. And our Kovacs reagent is there to detect the presence of indole. And so we have to add 7 drops to each tube. Now to the MRVP broth, notice that it's MRVP broth. And remember I said that this is going to be used for both an MR test and a VP test. So what we have to do is we have to take this broth and using a transfer pipette, we would transfer 1 milliliter of this broth. So this is already grown with E. coli. And we would transfer 1 milliliter to one tube and then another milliliter to a second tube. So I would have 2 tubes that have my E. coli in it that is already grown. And I would label one ECMR. This tube would get the methyl red reagent added. And I would label the second tube EC and VP. That would get the VP reagents added to it. And I would repeat the same thing for the EA. I would also separate that into 2 tubes. One is going to have methyl red, one is going to have VP. So to the tubes that are labeled MR, methyl red, you're going to add 3 drops of the methyl red reagent. To the tubes that are labeled VP, you would add 15 drops of VPA and then 5 drops of VPB. So there's 2 reagents to add to the VP test. You have to add A first and then B. And for the VP test, the readout can take up to 60 minutes. So you have to wait to see if you can see the color change. So it's not an instant readout like an MR test would be. So this table here on the bottom is just a place for once you start watching the videos, if I were you, I would go back and summarize what color or what result is a positive for the indole and what color or what result is a negative in the indole test. Same thing for MR, same thing for VP, same thing for citrate. So once you watch the video, I would fill in this table to have one place that tells you what the positive and the negative looks like. So I have a video now that I'm going to show you that demonstrates the procedure of doing the readout. And then we'll talk about the different tests, the biochemical sheets, and also what the readout tells us. So here's the video showing you how to do the readout. Okay, so this is the second part of this experiment, doing the readout day. So what you do is after the 48 hours, you're gonna have your 3 tubes for each organism. So these are my 3 E. coli tubes. I have one with the green cap. Remember, this is my tryptophan broth. This is looking for my indole production. I have my red cap. This is my MR-VP broth. This is used for both the MR and the VP test. And then my citrate slant. So what I need to do is that for my tryptophan broth, notice that there is some turbidity, there is some cloudiness. So that tells me that the bacteria grew. And for my tryptophan broth, I need to add my Kovacs reagent. So here's my Kovacs reagent, and I need to add 7 drops of Kovacs. So I'm going to take my Kovacs, and I'm going to add my 7 drops. 1, 2, 3, 4, 5, 6, 7. And I'm going to wait to see if a pink ring develops on the top. So I'm done with my indole test for now. So I'm going to move away my Kovacs. And now I need to do my MR-VP broth. So here's my MR-VP broth. Again, we can see turbidity. It grew. It's cloudy. But this broth is going to be used into 2 tubes. So notice what I have is I have 1 tube that I labeled EC-MR. This is my methyl red tube. And I have another tube that I labeled EC and VP. This is my Vogue's Proskauer tube. So what I need to do, I'm going to move this one aside for a second, is I need to use a transfer pipette, and I need to transfer 1 milliliter of this broth to each of these 2 tubes. So to do that, I'm going to take my transfer pipette. This is sterile, it's individually wrapped, and I'm going to just peel this open. Now, on these transfer pipettes, there's a little line at the top, and this little line at the top is how we know where 1 milliliter is. I need 1 milliliter of this into each tube. So I need to make sure that I don't contaminate my dropper, so I'm careful when I take it out. And I'm going to, let me just vortex to mix before I go in there. And I'm going to take the cap off. And I'm going to draw up to that line and then add it to the tube. And then same thing, draw up to the line and then add it in. So there is some extra broth in there. That's to be expected. We can just put that aside in case we need it. I put the transfer pipette back in the wrapper so that I don't set it on the desk and get bacteria on the desk. So it's in the wrapper. So now I have my 2 tubes. I have my MR and my VP. So to my methyl red tube, I'm going to add 3 drops of the methyl red reagent. So I'm going to take this and 1, 2, 3. To the VP tube, I need VPA and VPB. So this is my VPB. So we can see this is my VPB. This is my VPA. Now, I don't want to get these reagents on my hand, so I'm not going to hold my tube as I add the drops. I am, however, just going to, let me move this out of the way. I'm just going to add 15 drops of VPA to the tube. So I'm just going to do my dropper. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. And you can smell that this is acetic acid. So you don't want to get this on your skin, which is why I do it with the dropper there. So 15 drops of VPA. Now I'm going to use my VPB and I'm going to add 5 drops. 1, 2, 3, 4, 5. And then I'm going to let those sit. So my methyl red, the readout can be done right away. VP though can take up to an hour. So the VP has to sit for a little bit before you can do the readout. The MR, generally you can do the readout right away. The citrate, the readout is looking at the slant. I'll talk about this when I analyze the data. And the tryptophan could be analyzed right away as well. And so this is how we do our readout for our IMViC. So now let's talk about the readout for our IMViC. And so the first letter in our IMViC test is I, and the I stands for the indole production test. And the purpose of this test is to determine whether bacteria can produce indole from tryptophan. Tryptophan is an amino acid. And so we have our amino acid substrate. So here is our amino group, our carboxyl group, our central carbon, our hydrogen, and our R group. And so for our R group for tryptophan, this is what it looks like. So we're working with an amino acid substrate. That's what needs to go into this reaction. And we are going to hydrolyze or break down tryptophan. So again, if we're going to break something down, this requires hydrolysis, right? Hydro, water, lysis, breaking. So we're going to use water to break down this tryptophan. And the enzyme that does this is called tryptophanase. And tryptophanase is an endoenzyme. And what that means is that the enzyme is produced within the bacterial cell and it stays in the bacterial cell. So it's not an enzyme that is secreted out of the cell, it actually stays within the bacteria. So our substrate is tryptophan. Our enzyme is our tryptophanase. And so you want to write on your biochemical sheet both the name and the fact that it's an endoenzyme. Later on, you will see some examples of what we call exoenzymes, which are enzymes that are secreted out of the cell. And there's a reason that those enzymes are secreted out. So you want to start studying these now, which ones are endoenzymes and which ones are exoenzymes. So in addition to the name, you should know that it's an endoenzyme. So what's going to happen in tryptophanase is it's actually going to take tryptophan and it's going to break it into 3 parts. So the first is that the amino group is going to be broken off, and that will give rise to one product, which is ammonia. Then we have this R group portion that gets broken off, and that is going to give us indole. So indole is our second product. And then to the remaining part for tryptophan, that is going to give rise to pyruvate. So we take this amino acid substrate and we break it into 3 parts. So we end up with 3 products from this reaction. We have indole, we have ammonia, and we have pyruvate. And the purpose of doing this is that when we use that amino acid, we can make pyruvate. Pyruvate is a product that you guys will learn about when we learn about metabolism. Pyruvate is a product of glycolysis, one of the first steps in respiration. And that pyruvate then can go on and be used to get broken down further in respiration, or it could be used for fermentation. And so this is basically a source of energy for the cell to make ATP. And so we have our substrate, our tryptophan. We have our enzyme, which is our tryptophanase. And it's going to break down the tryptophan into 3 products. indole, ammonia, and pyruvate. So now we need to talk about what does a positive test look like? What does a negative test look like? Because we want to know, do bacteria hydrolyze or break down the tryptophan? So to determine if this reaction occurs, to our tryptone broth, remember that this tube is a tryptone broth, it's the one with the green cap, we added 7 drops of Kovacs reagent. And the Kovacs reagent itself is a pale yellow. And the purpose of the Kovacs is that it detects the presence of indole specifically. So it will change color if indole is produced. So this Kovacs reagent will allow us to determine if this reaction occurs, because if indole is produced, the reagent is going to change color. So a negative in this test, meaning that indole is not produced, the negative in this test will stay yellow. And what that tells us is that no indole was produced. Because if indole was present, the Kovacs would change color. But if it stays yellow, it didn't change color, and therefore indole was not produced. The positive is going to turn pink-red. And if it turns pink-red, that tells us that indole is present. Because remember, Kovacs starts out that pale yellow color. So if the color changes, the only way that the color changes is if indole is present. So if we see pink on the tube, on the top of the tube, that tells us that indole is present. And what that tells us ultimately is that bacteria have this tryptophanase, they make this enzyme, which allows them to do this reaction. Now, in terms of our biochemical tests, sometimes students are wondering, well, why do we need to know about whether or not bacteria possess tryptophanase? Like, why do I need to know if bacteria produce that enzyme? The answer is that based on these results in the IMViC, it helps us narrow down which bacteria we have. And so that is going to be useful when you guys start working on your unknown project, where we give you unknown bacteria. And by process of elimination, you have to figure out what bacteria you have. One of the tests you're going to have to do is an IMViC, because once you get your results of your IMViC, that will narrow down what bacteria you would have in your experiment. So these biochemical tests are basically used to help identify an unknown bacteria. So in this case, we are looking at, do bacteria produce tryptophanase? And there are some bacteria that we know that produce that enzyme and others that don't. So now let's talk about why we can't use a pH indicator as a readout, right? You guys have learned about your phenyl red broth. You learned about the OF test. Those readouts were done using a pH indicator, right? Phenyl red used phenyl red, and the OF test used bromothymol blue. And those pH indicators change colors depending on the pH. However, in this experiment, we can't do that. We can't use a pH indicator. So on your biochemical sheet under pH indicator, you're going to put Na, not applicable. There is no pH indicator. And that's because if we look at our products, notice we have our 3 products. We have indole, we have ammonia, and we have pyruvate. Pyruvate is also known as pyruvic acid. Now, in terms of ammonia, if you know anything about ammonia, what is ammonia like in terms of its pH? And the answer is that ammonia is alkaline. It raises the pH. So we have ammonia, which is a base, and we have pyruvic acid or pyruvate, which is an acid. So notice that these products produce a base and an acid. And so are you going to see a net pH change? And the answer is no, they're going to cancel each other out. So ammonia is a base and pyruvate is an acid, so they will cancel each other out. And so we can't use a pH indicator because those products will not allow the pH to go up or down, because one is going to raise the pH and the other is going to lower it, and they're going to cancel each other out, and you can't detect a change in pH. So instead, in this test, we don't use a pH indicator, we use a reagent. And the reagent in this case is going to be the Kovacs. It's going to detect the presence of indole. Now, you might wonder, well, what really is a reagent? A reagent is typically something that we add after the incubation period. So notice that when we do this, we inoculate these tubes with bacteria. We put them in the incubator for 48 hours and we grow them at 37 degrees Celsius. So those tubes had grown for 37 degrees Celsius for 48 hours. So what happens then is that during that time, they grew, and then we don't have anything in there to detect if the reaction occurs. So that's why once the reaction, or once the incubation has gone for 48 hours, then we can add the reagent, and the reagent then will detect if indole is produced. So a reagent is typically something that we're going to add after the bacteria has already been grown. So that's why this is referred to as the reagent. Again, it's not a pH indicator because it's not changing color based on pH. It's changing color dependent on a specific product, and that product is indole. And so this is going to be our indole production test. So if we see pink red, that is a positive. If it stays yellow, that's a negative. So let's look at, for our 2 bacteria that we tested, let's see what our results were. So here are the results of this experiment. So here on the left, we have our E. coli, which was our EC. On the right, we have our Enterobacter aerogenes, which is our EA. So notice that for the E. coli, we see this pinkish red ring at the top. But for the EA, that is not present. So what that tells us is that E. coli is positive for indole production. E. coli possesses the tryptophanase. They make that enzyme, and they're able to take that tryptophan in the media, and they're able to convert the tryptophan into indole. And the indole is going to react with the Kovacs, and it causes the color change to the pink red. In the case of EA, Enterobacter aerogenes, that is a negative result. Notice that on the top, the reagent is yellow. And so that's the color of the reagent when it started, which means that there was no indole being detected. And that's why the tube stayed yellow, because there was no indole produced, which means that Enterobacter aerogenes does not produce tryptophanase. It doesn't make the enzyme, and therefore does not produce indole. And so these would be our results for the indole production test. This is the first letter in the IMViC. So now let's look at our MRVP. So for our MRVP, the purpose of this test is to determine whether bacteria ferment glucose into mixed acids or acetoin. So the MRVP is intertwined in that it's the same broth, but we're going to separate that into 2 tubes. To one tube, we're going to add the methyl red reagents. To the other tube, we're going to add the Voges-Proskauer reagents. So for both tests, we start with glucose. Glucose is going to be our substrate. It's our sugar. We're going to see if bacteria can metabolize or ferment the glucose, and whether they produce stable mixed acids or whether they produce acetoin. So for both of these tests, again, you want to have a separate biochemical sheet for methyl red and a separate one for Voges-Proskauer. So my substrate is the same. My substrate is glucose. The enzymes, you're just going to write various endoenzymes. This is not detecting any one particular enzyme. It's actually a collection of enzymes. It's a bunch of enzymes that are involved in these pathways. So that's why, just like with fennel red broth and with the OF test, you also wrote various endoenzymes because there are a lot of enzymes involved in those pathways. So our substrate is glucose. For both, our enzymes are just various endoenzymes. In the case of organisms that are methyl red positive, they will produce stable mixed acids. So they produce stable mixed acids. And what that means is that they produce a variety of acids which will all lower the pH. And so when the pH gets below 4.4, if we add our 3 drops of our methyl red reagent, then it will detect this change in pH, because methyl red is both a reagent and a pH indicator in this test. The reason it's also a reagent is because it's added after the incubation period, not like for fennel red where the pH indicator was already in the media. So in this case, for the methyl red is the reagent. We're adding it after incubation, and it's also a pH indicator because it will change color dependent on the pH. When the pH gets below 4.4, the color is going to change. And so the color will change if stable mixed acids were produced. However, some bacteria don't produce stable mixed acids. Instead, they do a different pathway. And in this different pathway, they will produce a product that is called acetoin. Now, organisms that produce acetoin, initially, they will ferment the glucose into acids. So we can't do this readout at 24 hours because at 24 hours, the bacteria have not had enough time to produce that acetoin. The readout has to be done at 48 hours. So acetoin is going to be an alkaline product, meaning it's going to increase the pH. In the case of the acetoin, we detect it by the VP reagents. There are 2 reagents. We have VPA, which we add 15 drops. We have VPB, which we add 5 drops. And then if we see a color change, that would tell us that bacteria produced acetoin. So in the case of methyl red, the positive test is going to be if it turns pink red, which indicates that the pH decreased. So again, with the methyl red, when the pH gets below 4.4, it's going to change, it's going to turn pink red. So if that tube is pink red after the addition of the methyl red, that tells you that the pH is less than 4.4 and that acids were produced. Now, notice that the methyl red is red when acidic. Remember, I've been telling you that throughout the semester, when we look at our pH indicators, there's kind of a go-to color in terms of acidic pH. And normally I said that the acidic pH is going to be yellow. So that's true for fennel red, that is true for bromothymol blue, that is true for bromocresol purple. In all of those 3 cases, yellow is going to be acidic. This methyl red is the exception to that rule. Methyl red is red when acidic. So you can think of it this way, that methyl red is the opposite of phenol red. They're completely the opposite, right? Because phenol red turns red or pink when it's alkaline. Methyl red turns red or pink if the pH is acidic. So methyl red is the opposite of phenol red. So methyl red will turn pink red if the pH decreases, which would indicate a positive for bacteria fermenting glucose into stable mixed acids. In the case of the VP, the Voges Proskauer, that is going to turn pink red if acetoin is present. So only if acetoin is present will it turn red. So notice something interesting. In the first 3 tests, indole, methyl red, Voges Proskauer, all 3, pink red is positive. So that makes it a lot easier when you're studying that you can remember for the first 3 letters of the IMViC, pink red is going to be the positive. If you see that pink red, that tells you that that test is positive. Bacteria produce those products. So this is what we do in our MRVP. We want to see, do bacteria produce stable mixed acids or do they produce acetoin? And so that's what we're going to be looking for in this test. So now we need to understand about why the timing of this experiment is so critical. So when we start out, our pH is neutral. The pH starts at about 7 because remember that most bacteria are neutrophiles. They prefer a neutral pH. So the pH is going to start out neutral. If the organism is MR positive or methyl red positive, that means that they will produce stable mixed acids. And so notice that because they produce stable mixed acids, it is going to lower the pH, right? The pH is going to go down. It's going to become more acidic, and it's going to stay low because they're stable mixed acids. So it lowers the pH and it stays low. And so when we add the methyl red reagent, it's going to turn red because the pH is below 4.4. So that's for the MR organisms. Now, notice something different about VP positive organisms. And that is, remember that initially this bacteria is going to produce acids. So the pH is going to start to go down first. If you were to do this experiment and do the readout at 24 hours, that is not likely going to give you the correct result for 2 reasons. If that pH gets low enough, this tube that actually is a VP positive organism, at 24 hours, this tube is going to be acidic, which means that it might be methyl red positive if you added the methyl red. And it would be VP negative because there's not enough time for acetoin to be produced. So notice that we cannot do this readout at 24 hours. If we do that, we could get the wrong result. We would say that this organism, as a result, based on the readout, that the organism appears to be MR positive, but they're not. It's just that you did your test too early. So you have to wait the 48 hours. So in a VP positive organism, it's going to produce acids initially and lower the pH. And then it's going to take those acids and it's then going to produce the acetoin. And the acetoin is going to raise the pH because that product is alkaline. And so the VP reagents are going to detect the presence of the acetoin. So now there's a question on your question set that I wanted to review with you. And that is, notice this question was one that has a star, which tells you that it's a question that's a little more challenging. So notice that the question asks, so for question number 90, why are organisms that are MR positive usually VP negative and VP positive organisms are usually MR negative? So what that means is that typically bacteria will only test positive for one or the other. Think about why that is. So let's go through the example. Let's start with a VP positive organism. So if you take a broth, right? And that broth, you've grown your bacteria for 48 hours. You then take that broth and you split it into 2 tubes, right? So you're going to take this and you're going to split it into 2 tubes. To one tube, you're going to add methyl red. To the second tube, you're going to add the VP reagent. Now, if the organism is VP positive, right? The only way that you get an organism that is VP positive is if acetoin is produced, because the VP reagents react with the acetoin specifically. So any organism that is VP positive, it means that they have produced the acetoin. Now, if they produce the acetoin, is it likely that the pH is going to be acidic? And the answer is no. It's not also going to be acidic, because if it produced the acetoin, that's going to raise the pH. That's why it's VP positive, because of the production of the acetoin. But when the pH is up, it should then be negative with the methyl red, because the methyl red is detecting acids. The acetoin is going to raise the pH. So there's a reason why they're usually one or the other. So I wrote, if an organism is VP positive, then it means that they produce acetoin, which raises the pH. Therefore, it shouldn't test positive for MR2, which detects a low pH, right? And so they're usually not both. They're usually one or the other. They're usually VP positive or they are MR positive. Not both, because they can't produce acetoin, which is alkaline, but then also be positive for acids. There are a few rare exceptions to that. However, for the general rule, that's not the case. So they're one or the other. Now, another critical thinking question. Would this test work if you have an organism that does not metabolize the glucose? Meaning, let's say you had an organism that is non-saccharolytic, that doesn't use sugar. And the answer is this test won't work, because in this test, we need to ferment the glucose for us to either have the acids or the acetoin. So if the organism can't ferment the glucose, then it's not going to be positive for either. Similarly, if you made MRVP broth and you forgot to add the sugar, you forgot to add the glucose when making the media, this test would not work, because if we don't have a substrate, we can't produce those products. So just some things to kind of think about when you're thinking about your MRVP. This test will work, or can work, if the organism is able to metabolize the glucose. And this test is usually going to be positive for one or the other, but not both. Because again, they're going to have opposite pH changes, so it's not likely that you're going to be able to detect both. So what we did for this test is that we inoculated bacteria into an MRVP broth. And then we incubate the bacteria at 37 degrees Celsius for 48 hours. So our bacteria grew. This tube has turbidity. And so what I did was then I took this tube, this MRVP broth, and I transferred 1 milliliter into 2 separate tubes. And so I separated it into 2 tubes. I added to 1 tube 3 drops of methyl red, and then we can do the readout right away. To the second tube that I transferred 1 milliliter, I'm going to add 15 drops of VPA and mix. I'm going to add 5 drops of VPB and mix. But once I mix initially, I do not want to agitate that tube anymore. I need to leave it alone after I add the reagents. So you're not going to continue to vortex throughout the experiment. You're just going to mix when you add the reagents and then let the tube be. You're not going to shake the tube. You're not going to vortex the tube. For the VP test, it can go up to an hour before you see the color change. And so you would just check it every so often to see if the color changed. If we see pink red, that is going to be positive for methyl red. And the methyl red, again, the color change is going to happen instantly. If the color does not turn red, that means that the organism is MR negative. It doesn't do mixed acid fermentation because it didn't produce acids to lower the pH. For the VP, again, the reagents, the VPA and the VPB, they're going to detect the presence of acetoin specifically. If acetoin is present, it's going to turn pink red. But that pink red could take up to an hour. If, on the other hand, we add the VP reagents and we wait an hour and we still don't see any pink red, if that's the case, then the organism is VP negative. They don't produce acetoin. So this is our result. Now, one of the things you're going to see in the next slide, this image in the lab book shows it being red all throughout. In actuality, what you want to realize is that it's actually not usually red throughout. Usually the VP reagents will form a ring of red at the top. Just like you just saw for the indole production test, when we add the Kovacs, the pink red is not all throughout the tube. It's only at the top. Same thing for the VP reagents. You're going to see a ring at the top. It's not red all throughout like it's shown in this picture. Methyl red is. Methyl red, the whole tube turns red. But for VP, you're just going to end up with a ring along the top. If you see that red or pink ring, that is positive for VP. So let's look at the results. So when we are looking at our mixed acid fermentation, the MR part of this experiment. So for the MR, the bacteria is going to go through glycolysis, glyco, sugar, lysis breaking. That is where we take our 6-carbon glucose and we chop it into 2 3-carbon pyruvates. Now, when we do this, there will be various enzymes. So again, that's the various endoenzymes. And they will produce a variety of different acids. Lactate, which is also known as lactic acid. We have succinate or succinic acid. There are a bunch of different acids that come into play or that get produced if the organism does a mixed acid fermentation. So if they do a mixed acid fermentation, they're going to produce a variety of stable mixed acids, products that are going to lower the pH. So if the organism is MR positive, that means that if we see that pink red, that means that acids were produced and that the pH was less than 4.4. Because again, we don't see that pink red unless the pH gets below 4.4. So in this case, the methyl red test is positive because it turns red, which tells us that the pH is below 4.4, which tells us that stable mixed acids were produced. If the tube is any color other than red, so orange would not count, yellow would not count, it has to be pink red to be considered positive for MR. So if that tube is yellow, then we would record that as MR negative. It's methyl red negative. It does not produce stable mixed acids. So in that case, the acids are not produced. The pH is above 4.4, which is why the tube is not red. So in our MR test, red is positive. It's detecting a pH change. It's detecting the acids that are produced. MR negative is going to be when mixed acids are not produced. So meaning that the pH did not decrease because the bacteria did not produce the stable mixed acids. So for our VP test is a little bit different. So again, we still have the same beginning. We still have glycolysis, where we're going to take this 6-carbon glucose. This is just showing glucose in its ring form. And in glycolysis, we're going to take that glucose, chop it into 2 3-carbon pyruvates. So notice that this beginning part is very much the same. It's after that that the process becomes different. And so in a VP positive organism, a VP positive organism is going to produce acetoin. And then that acetoin can be used to produce 2 other products, which are the diacetyl and the 2,3-butanediol. These 2 products come from acetoin. Now, in our VP test, we are detecting the presence of the acetoin. The VP reagents they're not pH indicators. They don't change color depending on pH. They change color if acetoin is present. So here are my VP reagents. So we have our VPA, which is our alpha naphthol. That is our VP reagent A. And so we add 15 drops of the reagent A first. And so if we have acetoin present, and then we add the VPB, which is going to be our potassium hydroxide, and notice it needs oxygen, which is why you should mix. So once you add the VPB, right, that is going to then react with the VPA and the acetoin. The acetoin is going to produce this diacetyl, and there's going to be this guanidine in the peptones that are in the media, and this is going to cause a red color change. So for this one, you don't need to know all the specifics of this color change here. What you really want to understand is that that pink-red color comes from acetoin being produced. These VP reagents will detect if acetoin is produced. If acetoin is produced, we will get a red color. So if we see that it is red, what that means if it's red is that the VP positive organism is telling us that the acetoin is detected. So that is why we see this red. So acetoin is detected. VP negative is if it's not red. So if it's not red, acetoin is not detected. So that means that the acetoin is not produced. Therefore, the organism does not produce acetoin. And so they are VP negative. Notice that the reagents themselves give off this kind of coppery color. That is normal. That is not positive. Positive has to be this distinct, like bright pink red color. So again, like methyl red, red is positive for the VP. So in this case, if it turns red, that means that acetoin was detected and that organism is VP positive. If the tube is not red, then that means that the acetoin was not detected and this organism would be VP negative. It does not produce acetoin. So let's look at our results for this experiment for the organisms that we're testing. So here are the results for the methyl red. So we have E. coli and we have our Enterobacter aerogenes. Notice that for E. coli, it is pink for the methyl red. What does that tell us about the E. coli? Is it positive or negative for methyl red? The answer is it is positive for methyl red, because if it turned red, what that means is that the pH decreased. It's below 4.4, which causes the methyl red to turn red. So that's going to be our positive. Notice for the Enterobacter aerogenes, we see not red, so that is negative. The organism is not capable of doing a mixed acid fermentation. So Enterobacter aerogenes does not do mixed acid fermentation. Now let's look at the results for the VP. So again, we have E. coli on the left, and we have our Enterobacter aerogenes on the right. Notice for this time, if you look at E. coli at the very top, it has this very faint pink, almost kind of coppery color. Again, that is not the positive. This is not positive. That's just the reagents reacting with one another. That is not a positive. So if we see this result for E. coli would be negative for VP because it's not pink red, so there is no acetoin produced. But we see that pink red ring at the top of the Enterobacter aerogenes, which tells us that EA is positive for VP. Now notice something, and that is that E. coli is MR positive, but VP negative. Enterobacter aerogenes is MR negative, but VP positive. So again, you can see that they're not positive for both. Each organism is typically going to only be positive for one, because either they produce stable mixed acids, which would lower the pH and would be MR positive, or they produce acetoin, in which case the pH would go up, and so they would test positive for VP, but not for methyl red. And so these are the results that you would see for your MRVP test. So again, your MRVP is 2 tests in one. It's our methyl red and our Voges-Proskauer. Voges-Proskauer is detecting if acetoin is produced. The MR is detecting if stable mixed acids are produced. So now we're going to move on to our last part of our IMViC, which is the C. And the C again stands for citrate utilization test. And the purpose of this test is to determine whether bacteria can utilize citrate as a sole carbon source, meaning that the citrate is the only source of carbon. So the name of our media for this experiment is referred to as Simmons citrate agar. The recipe for this is on page 340 of your lab book. And so let's discuss what each of these ingredients are used in the media for. So we'll start with the sodium citrate. So the sodium citrate is our sole source of carbon. It's the only carbon in the entire media. Organisms need carbon to make all macromolecules. So what that means is that organisms have to be able to take in carbon in some form to be able to survive, because they need that carbon to make their macromolecules, to make their proteins, to make their lipids, to make their nucleic acids, right? All of our macromolecules contain carbon. Notice when you look at this media, you'll notice something that is missing from this media. And that is there are no peptones. There are no, yeast extract or beef extract or pancreatic digest of casein or pancreatic digest of gelatin. None of those ingredients are in this media. So what do we call this type of media that does not contain peptones? It does not contain ingredients that vary from batch to batch. And the answer is this type of media is referred to as defined media, meaning we know every ingredient that's in there and it's not going to vary from batch to batch. So what that means is that if this is the only source of carbon, right? If we give them the sodium citrate and they cannot use the citrate, can they grow? And the answer is no. They cannot grow if they cannot use the citrate, because again, all organisms have to take in a source of carbon. If they can't use the citrate, they have no source of carbon and therefore they can't grow. So this test is a little bit different. Remember in our fennel red broth test, I said that if you get no growth would be considered inconclusive or no result, because we gave them multiple food sources. We gave them sugar, we gave them peptones. There were multiple sources of carbon, and so bacteria should have been able to use something in the fennel red broth. The citrate test is different because this is a defined test and the only source of carbon is going to be the citrate. Bacteria that don't use citrate cannot grow. So again, this is going to be different in that no growth is a negative in this test. And I'll come back to that in a minute. So we have our sodium citrate. This is our sole source of carbon. Then we have our ammonium dihydrogen phosphate. And our ammonium dihydrogen phosphate, or our ammonium phosphate for short, is our source of nitrogen. So if you think about elements that organisms need to grow, they need carbon to make all macromolecules. They need nitrogen because nitrogen, remember, is used to make nucleic acids, so to make DNA and RNA. Nitrogen is also needed to make proteins. So for a bacteria to build their own proteins, to make their enzymes, they will need to take in a form of nitrogen. So the nitrogen that they take comes from this ammonium phosphate. Normally in the media, the nitrogen comes from peptones. We don't have peptones in this media, so we have to give them a source of nitrogen. And in this case, our source of nitrogen is the ammonium phosphate. So if the organism can use citrate as their sole carbon source, so if they're able to utilize citrate, then they must grow, right? So if they use the citrate, they will grow. And if they grow, they also need to take in a source of nitrogen, which means that they will utilize the ammonium phosphate. And when they break down ammonium phosphate, they will produce basic products, which will raise the pH. So in this test, in our citrate utilization test, this is what we call an indirect test. We are not detecting the products of citrate metabolism directly. We are not detecting any product of citrate itself. What we are instead doing is that if bacteria use the citrate as their carbon source, then they must also use the nitrogen source, and that nitrogen source is what's going to cause the pH to change. So again, this is an indirect test. We then have our dipotassium phosphate. That is our buffer. It's used to keep the pH constant. We have our sodium chloride, which remember is there for salt balance, right? So to make it so that too much water doesn't go into the bacteria, too much water doesn't come out. So we need the salt balance to be correct. We have our agar. Our agar is our solidifying agent because remember that the citrate agar is a slant. So we need to have agar to make it solid. And then we have our bromothymol blue, which remember is a pH indicator. And remember that the colors for bromothymol blue, again, the name, the blue, tells you the alkaline color. And remember we said that yellow is acidic. So the yellow is going to be acidic for all of our pH indicators except methyl red. That's the only exception. So for bromothymol blue, yellow is acidic. If blue is basic, what color is neutral going to be? Well, neutral is going to be between blue and yellow, which if you've painted, right, if you mix blue and yellow together, you get green. So green is our neutral pH and blue is our alkaline pH. So remember that when I inoculated the citrate slant, the slant started out green because most bacteria are neutrophiles. They grow best at a neutral pH. So if they grow best at a neutral pH, we start with the pH being neutral. So the pH in the tube is green when we start. If bacteria utilize the citrate, they will produce alkaline products, which will then turn the bromothymol blue blue. And so let's take a look at this. So this is our reaction for our citrate utilization test. So in this test, we have our citrate. Our citrate is our sole carbon source. It's the only source of carbon in the media. We also have a second substrate. So there's actually going to be two substrates listed for this test. The second substrate is the ammonium phosphate. This is going to be the substrate that eventually leads to the readout. Now, the citrate part, so metabolizing the citrate is going to use two enzymes that you should know. We have our citrate permease. That is an endoenzyme. It's actually an enzyme used to transport citrate into the cell. That's what it's called, permease, to get citrate in. It's a transport enzyme. And if the citrate gets in, bacteria also need to possess this citrate lyase, which will allow it to break down. Now, in the citrate metabolism, there are no products that we can detect directly. So what happens in this experiment is not that we're detecting products from citrate metabolism directly. We again are doing this indirectly. If bacteria use citrate, right, if they're able to utilize the citrate, they will grow. If they grow, right, they also need to then use a source of nitrogen, because they need that source of nitrogen to make their nucleic acids and to make their proteins. So if the organism is positive for citrate metabolism, that means that the organism also needs to utilize the ammonium phosphate. You don't need to know enzymes for this part of the pathway, so don't worry about those enzymes. But basically, if bacteria grow, they will use the ammonium phosphate as their source of nitrogen. And if they use it as their source of nitrogen, we get two products. Our products are going to be the ammonia, the NH3. This is not plus, meaning a charge on ammonia. Ammonium, which is NH4, which would be positive. Ammonia, which is NH3, is not positive. This positive here, this plus that's here, just is another word for and in this case. So we have two products. We have our ammonia, our NH3, and we have our ammonium hydroxide, so our NH4OH. These are the two products. These, again, are indirect products of citrate metabolism. So, again, these are not produced from citrate itself. They are only produced if bacteria use citrate, they will grow. If they grow, they must also utilize the ammonium phosphate. And when they use the ammonium phosphate, they produce these alkaline products. These alkaline products are going to raise the pH, and it's going to cause the bromothymol blue to turn blue. So in our positive test on our citrate utilization, blue, if you see blue, that is definitely a positive, because the only way that you get a pH change is if the bacteria grew. If the bacteria grew, that means that they had to utilize the citrate. So a blue is positive, and even with a small amount. Or there's another positive that could happen, and that is green with growth. So let's say we did this and we got a tube that was green, but we could see bacteria growing on the slant. If we see bacteria growing on the slant, that has to be a positive, because the only way that bacteria can grow at all is if they use citrate. So if we see growth, that is going to be a positive. Now you might wonder, well, why didn't it turn blue? Well, it didn't turn blue because maybe it needs a little bit longer to produce enough of the alkaline products to cause the pH to change enough for it to turn blue. So we're not as concerned in this case as much with the blue. The blue tells you it is positive for sure. Or if you see green with growth, that is still positive, because again, the only way bacteria grow is if they can utilize the citrate, right? So if they utilize the citrate, they can grow. So green with growth is still considered a positive. That is still a positive test because the bacteria had to utilize the citrate in order to grow. The negative test is only green with no growth. So green with no growth is our true negative, because bacteria cannot grow if they can't utilize the citrate. So again, this is different than our fennel red broth. Fennel red broth, remember we said no growth is inconclusive. In our citrate test, no growth is the negative, because citrate is a defined media. It only has one carbon source, and if bacteria don't use citrate, so if they don't utilize the citrate, then they cannot grow. So our true negative in this case is going to be green with no growth. Now, there's a couple of keys when setting up this experiment to make sure that your results are correct. One has to do with the way that we inoculate this slant. You might recall that I said that when we inoculated this slant, I inoculated it using a needle. The reason I inoculated with a needle is because I wanted to be sure to not transfer too much bacteria. Because if I use a loop and I pick up a lot of bacteria and I do my zigzag inoculation, if I transferred a lot of bacteria, it might give off the false impression that the bacteria grew, when in fact it didn't. You just transferred too much. So to avoid transferring too much bacteria, we use a needle when we inoculate this. We still do the zigzag inoculation, but we inoculate with the needle so we don't transfer too much. So that way, if we see growth, right, if we see actual growth, that would be considered a positive. The other key when doing this, and I showed this in the video, these slants are screw tops. If I were to accidentally put my lid on too tight, if I screwed the lid all the way on and air can't get in. If air can't get in, the bacteria can't grow, whether or not they utilize the citrate at all. So then we would have to repeat our experiment. Like if I were to get my can out and realize that I put the cap on too tight, my result is meaningless at that point. I can't make a conclusion because if air didn't get in, then the bacteria couldn't grow. And so you would get this false negative. It would appear negative when in fact it's just simply because you didn't loosen the cap. So the way that we set this up is very important to make sure that we get the correct readout. We need to make sure that the cap is loose, and we need to inoculate with a needle so that we don't transfer too much bacteria. So let's look at what our results would look like for our experiment. So first, the blue, right? Any blue would be considered positive. Green with no growth would be my negative. So let's look at our organisms here. So what we have is on the left, we have our EC. So that's our E. coli. Notice that the slant is green with no growth. The Enterobacter aerogenes is blue, and you can see the zigzag slant, and it has bacteria on it. So the E. coli is going to be our negative, and our Enterobacter aerogenes is going to be our positive, because the blue and growth is a positive. E. coli did not grow, and the tube is green, which tells me that E. coli are negative for citrate utilization. So what I'm going to have you do is I've summarized and I put all of the pictures together on this one slide. And I want you to go through and indicate for each organism. So for E. coli, was it indole positive or negative? Was it MR positive or negative? Was it VP positive or negative? And was it citrate positive or negative? And then do the same with the EA. And so this will make sure that you understand the readout for these tests. And then when we have our Zoom, we can discuss those results to make sure that you interpreted this correctly. And so this is our IMViC. It's 4 tests in one. We have our I is our indole production, our M is our methyl red, our V is our Voges Proskauer. This little I again doesn't really stand for anything. It's just to make it easier to say. And then we have our C, which is for our citrate utilization. And so this is our IMViC test.